The overall goal of this procedure is to investigate the transport of charged and uncharged molecules across articular cartilage with the aid of experimental and numerical methods.
This method can help answer key questions related to physical properties of articular cartilage, such as diffusion coefficient and fixed charge density in different zones.
The main advantage of this technique is that it adopts a finite path experimental model based on microcomputed tomography of transport of radiopaque molecules.
To begin sample preparation, drill out an 8.5 mm diameter cylindrical osteochondral plug while spraying room temperature phosphate buffered saline, PBS, onto the drilling area. Insert the plug into an approximately 2.5 cm length of heat shrink tubing so that the bone layer is flush with the end, and then cover the cartilage surface with wet cotton. Then use a hot air gun to shrink the tubing around the sides of the plug. Load a finite volume of a 420 millimolar ioxaglate or iodixinol solution onto the cartilage surface inside the tubing to prepare a negatively charged or neutral solute bath respectively. Seal the open end of the tube with a cork. Place the sample with the corked end up on a sample holder mounted on the motorized stage of a micro-CT. Scan over a field of view containing the cartilage, subchondral plate, and finite bath. Repeat the scan periodically over the next